Okay, hello everybody. Thank you for attending. My name is Connor Secora, and the title of my project is Traumatic Brain Injury Exacerbates Alzheimer's Disease in the Retina. And I'd like to thank the many people that put so much hard work into this project. It definitely would not have been possible without everybody here. So I'd like to provide some background to help establish context for the study, starting with Alzheimer's disease, or AD. And AD is a progressive neurodegenerative disease that currently affects over 5 million Americans. This disease is characterized by the pathological aggregation of two proteins, amyloid beta, or A beta, which forms plaques, and tau protein, which forms neurofibrillary tangles, or NFTs. And both of these have a neurotoxic effect on the surrounding tissue, ultimately causing neuronal death and subsequent cognitive deficits. Interestingly, Alzheimer's pathology is known to manifest in the retina, and several visual defects accompany the progression of Alzheimer's. Traumatic brain injury, or TBI, is another disease that affects the central nervous system, resulting from direct or indirect trauma to the head, and has growing incident rates within the US over the past few decades. The progression of TBI includes an inflammatory response that recruits glial cells within the neural tissue, and typically also involves vascular injury that can include disruption of the blood-brain barrier. And TBI has been associated with a twofold increase in risk for developing Alzheimer's, thus linking these two prominent CNS-related maladies. So in light of these observations of Alzheimer's and TBI, we hypothesized that TBI exacerbates Alzheimer's histopathology within the retina. And part of the reason why we focus on the retina is due to the poorly understood mechanisms underlying the visual impairments related to Alzheimer's. And also the retina could be a useful biomarker for Alzheimer's diagnosis and assessment by providing an accessible and non-invasive means of analyzing neural histopathology. In order to test this hypothesis, we used a novel transgenic rat model of Alzheimer's and TBI in combination. And as we know, animal models are an essential tool to primary research by allowing us to perform controlled experiments that cannot be performed in humans. And to date, most of the Alzheimer's research has used mouse models, transgenic mouse models, which are incredibly useful, but unfortunately don't fully recapitulate human Alzheimer's pathology due to a natural resistance to forming NFTs. However, a recently developed Alzheimer's rat model has demonstrated the full spectrum of human Alzheimer's pathology with the expression of only two human mutant genes that predispose the rat to accumulations of A beta proteins. So this closer approximation to human pathology makes this model a promising tool to explore the correlation between TBI and Alzheimer's, which has not yet been done with this new model. And in this study, a TBI paradigm was incorporated by subjecting the rats to two events of unilateral controlled cortical impact using this apparatus shown on the left, or sorry, shown on the right. So there were four categories of experimental conditions as seen here. AD, or Alzheimer's transgenic rats compared to wild type, and then rats subjected to TBI compared to rats with a sham injury, meaning that the rats went through everything in the TBI protocol, including being anesthetized, everything except for the actual impact. And this created the four different groups of rats that we used for comparison. And the two CCIs, or sham injuries, were delivered at 12 months. And then the rats were euthanized at 16 months and prepared for histological examination. On the left, we could see a healthy rat retina stained with H&E, which shows the retina's highly organized appearance. We have three layers of nuclei separated by layers in between that are composed of the cellular extensions. And the graphs on the right, there's a lot of data here, but the graphs on the right basically indicate that we did not detect a significant difference in the thickness of these different layers between the four groups of rats. And this was surprising since many other studies have reported a thinning of these layers in Alzheimer's due to neuronal loss, primarily of the retinal ganglion cells that are mostly concentrated in the RGC layer. And since that neurodegeneration has been associated with ganglion cells, we quantify the number of these cells specifically using QCD as a marker, shown here in red. And we also tested for signs of cell death using cleaved caspase-3, shown here in green, which is a terminal effector of apoptosis. And as the graphs on the right indicate, we did not detect a significant difference in the number of ganglion cells between the different groups. And this finding, combined with a lack of differences in retinal layer thicknesses seen in that last slide, suggests that there was not a significant degree of neuronal loss observed in the retinas of these rats. However, we did notice a trend towards higher cleaved caspase-3 expression in the AD-TBI retinas, especially in blood vessel walls, as seen here, which may indicate early phases of neurodegeneration. We also examined the inflammatory responses within the retinas using glial fibrillary acidic protein, or GFAP, which is expressed in glial cells that have been activated as part of an inflammatory response. And the dominant glial cells within the retina are astrocytes within the nerve fiber layer, kind of shown up here, and then Mueller cells, which extend throughout the entire retina, as seen in these dendritic extensions. We therefore quantified the GFAP expression of these two different cell populations, shown in these two different graphs, and noted a significantly higher activation of Mueller glia in both AD retinas, indicating a heightened inflammatory response there. And then finally, we investigated the histopathology of Alzheimer's by quantifying the presence of A beta proteins using 4G8 antibodies in green. And unsurprisingly, since these rats are predisposed to accumulate these proteins, we detected a significantly higher expression of A beta in the two transgenic rat groups. In addition to the detection of just the A beta proteins, we analyzed the state of protein aggregation and plaque formation using a fluorescent dye called NIAID in red. And this has a high affinity for A beta fibrils and therefore highlights plaque-like A beta protein deposits of higher density. And interestingly, although both AD rat groups had similar counts of A beta protein deposits, a significant number of A beta plaques were only detected in the AD-TBI group, which supports our hypothesis. So in conclusion, we did not detect a significant difference in ganglion cell numbers, which may be due to the low power of the study or perhaps an indication of early stages of neurodegeneration. We did, however, find evidence of an increased inflammatory response in the retinas of Alzheimer's rats, and ultimately our findings of increased A beta plaque formations within the AD-TBI retina supports the hypothesis that TBI can indeed exacerbate AD histopathology. And this finding is important since it helps us evaluate the long-term effects of concussions in TBI. And these results merit further investigation using this AD-TBI model, such as the evaluation of functional differences in visual defects to correlate to the histopathology observed in the retina. It would also be fruitful to further correlate the retinal histopathology to the brain in order to better elucidate the relationship between Alzheimer's and TBI progression in the two different organs and further explore the diagnostic potential of the retina. Thank you for your time, and I'll open the floor for any questions if there are any.
Great, thank you, Connor. Great talk. It was really nice. Any questions for Connor?
I have a question. That was excellent, Connor. Really good job. And I know, just to promote your lab, that Dr. Vergara is looking for somebody else, anybody interested in continuing this project. Could you go back to the slide? What layer does the plaques tend to aggregate? Or did you find them in a certain layer?
That is a good question. They tended to present in the RGC layer where the retinal ganglion cells are, okay, in the ECM. We found some sparse plaques in the boundary of the internuclear layer as well, but they're predominantly in RGC layer and the interplexiform layer.
And did you look at optic nerve at all? You thought?
It's a good question. Not systematically.
Yeah.
We defined a couple of region of interests within particular areas that were radially displaced from the optic nerve, and we used those as representative areas of the entire retina, the posterior retina at least, but we haven't done any systematic investigation of the optic nerve yet.
So in the previous studies that you had looked at, did they use the same timeline, 12-month and 16-month? Like, what do you think would be the next step, to wait longer, or?
A few of those studies had similar timeframes, but the ones that reported the most significant differences went up to 27 months of age in the rats.
Oh, wow. Okay.
Yeah, very advanced stages of neurodegeneration.
So do you think the next step will be to wait longer and look, or what's your next, if you were to stay on, what would you do next?
I think there are so many different ways that we could continue this study. I think an interesting area to investigate would be looking at more the acute phase, to further differentiate maybe the mechanism of TBI and how it correlates to Alzheimer's. So we have a couple of time groups that are like 3 days post-injury, 30 days post-injury.
Oh, interesting.
So that could be interesting. It would also be interesting to see, investigate more of the long-term effects too.
Yeah.
If we look at like 20-month-old rats or something along those lines.
Yeah, it's a really nice study and an interesting approach to study the retina as sort of a window into the brain, which it is.
Yeah, yeah, exactly. And there are some studies being performed already to investigate some aspects of the histopathology using the retina, but there's still a lot of uncertainty on exactly what the relationship is, like how they're temporally integrated between the two, and how any differences in the phenotype to kind of heighten the diagnostic sensitivity. So a lot to be done, but very promising results so far.
Yeah, good work.
Great. Thank you.